afternoon, everyone, and good morning. And welcome to the second session of Digging Deep with Quantitative Plant Biology, the webinar series. If you don't already know, Quantitative Plant Biology is an open access journal published by Cambridge University Press in association with the John Innes Centre. We're delighted to bring you this webinar series thanks to our editors, authors, and the support of the John Innes Centre. Today, we're joined by special guest speaker, Satoru Tsugawa, and in a moment, I'll hand over to Olivier Hamant, the Editor-in-Chief of Quantitative Plant Biology, who will introduce today's session. Before we get started, I'd like to go over a few items so you know how to participate in today's event. You have all joined in listen-only mode, which means you'll be able to hear us but not able to speak to us. However, you will have the opportunity to submit chat questions by typing your questions into the Q&A of the control panel. You may send in your questions at any time during the webinar. We will collect these and address these during the Q&A session at the end of today's webinar. If we are unable to get through all the questions during the session, they will be included in the Q&A document that will be sent after the webinar. Should you experience any technical difficulties, please add this to the questions box and a member of the organising team will endeavour to help you. In the unlikely event we experience any issues, we will message the audience and restart the webinar. To summarise, you can submit your questions at any time and you'll also receive a survey at the end to know how useful you found it and another opportunity to ask questions. I would like to encourage all of you to use the Twitter hashtag QPB webinars to share your thoughts on the event and start meaningful conversations. So to kick things off, I'd like to invite Olivier Hamant, the Editor-in-Chief of Quantitative Plant Biology.
Thank you, Myra. So it's a pleasure to have everyone here today. So just a few words about the journal. So Quantitative Plant Biology is accompanying a revolution in plant science, which probably everyone in the room can experience, that plant science is becoming increasingly quantitative and interdisciplinary. I mean, it started probably with omics, but then it's went into more statistics, physics. And so this journal is going to support this effort and create a community around this revolution. And so today is a perfect example of this interdisciplinary field growing up. It's so part of the interdisciplinary field is the merging between physics and biology. And Satoru Tsugawa is a key example in this prospect, because Satoru is coming from physics and joined biology, and now is really a key figure in the quantitative plant biology. And so actually I should disclose that we worked together with Satoru a few years ago, and actually even more recently on microtubules. But today, Satoru will tell a very, very different story, which is key for plant science. It's about tropism in a way, and so this is something that started with Charles Darwin, and so it's really rooted in the history of science. And so this is a nice example where we see that quantitative plant biology can accompany this revolution one after another, and it's rooted in the history of plant science. So Satoru, it's a pleasure to have you today, and the screen is yours.
Okay. So can I share a screen?
Yeah, you can share the screen. Yeah.
Okay. And can you see the slide?
Perfect. Yep. All right.
Okay. So thank you very much, Olivier, for the introduction. And first of all, I'd like to express my deepest gratitude to the organizers, especially for Alison Paskins and Myra Carrigan, who have kindly invited me as a webinar speaker and prepared for this journal club. And also, I would express great appreciation to Olivier and Richard Morris, who have supported the publication process of this article. And my talk title is A Mathematical Model Explores the Contributions of Bending and Stretching Forces to Shoot Gravitatorism in Arabidopsis. And this work is a collaborative work with two physicists, Dr. Tomohiko Sano and Dr. Hiroyuki Shima, who are working on mechanical instability in elastic material and the bamboo mechanics, respectively. And the other collaborators are Dr. Mio Terao Morita, who is a well-known plant physiologist of shoot gravitropism, and Dr. Taku Demura, who is a specialist on genetic regulation of cell wall. And so thanks to great efforts of the excellent collaborators, this work could be achieved. And I would note that my current affiliation is Akita Prefecture University. So today I would present this from Akita, the small prefecture at the north side of main island in Japan. Okay, and here is today's outline. And the first 4 topics are introduction of myself and shoot gravitropism with data analysis and model analysis. Based on this, what I wanted to do was to make a correspondence between data and model to extract mechanics during shoot gravitropism. And finally, I would summarize the story and talk about some future perspectives. Okay, and to introduce myself, I have been working on plant morphogenesis with different scales and with different approaches. My major research topic at the student days was statistical mechanics in physics. So my research interest is basically physics in plants. But before going to physics, I first joined the quantitative biology laboratory to understand the real data. And in that project, I met with Olivier as a collaborative PI to investigate the CPAR developmental process. My contribution in the project was the quantification of microtubule and visualization of growth. Sorry, I will use this guy here. A visualization of growth in cell lineage. So please have a look at these works if you are interested. And after the first postdoc term, I joined the mathematical biology laboratory and worked on xylem pattern formation here. And to skip some details, we found that pit pattern in xylem cells can be explained by protein dynamics and showed a self-organization pattern in a specific parameter region in the model. Also in that term, I kept working on mathematical model of CPAR development and constructed a finite element method simulation. And in this model, I can modify the growth of CPAR spatio-temporally. So this color shows the spatio-temporal variation of growth. So red shows slow growth and blue shows fast growth. So using this type of model, we can discuss what happens to the macroscopic final shape based on the growth fluctuation at the macroscopic scale. And taking advantages of these collaborative works, I have recently started to work on physics based on both data and model. And one of the recent interesting topic was the corolla buckling. And in this paper, we discuss a specific mutant with folded petal in morning glory and extracted mechanical stress during development based on micro CT scan and the finite element method. And most recently, I'm working on shoot gravitropism, and I'd like to talk about this topic today. Okay, so then what is shoot gravitropism? The shoot gravitropism is an upward bending phenomenon against gravity of plant shoot. So if the shoot is inclined to be a horizontal orientation, the shoot gradually bends upward to the vertical orientation. Interestingly, this bending occurs without light. So the plant somehow knows their own position in darkness, which might be acquired in the evolutionary process. And the molecular mechanism of gravitropism is well studied so far. And the first trigger is thought to be the gravisensing by amyloplast, where the amyloplast in the endothelial cells move downward according to gravity. After the sensing process, the differential cell growth is induced by auxin localization, where the upper part with weak auxin transport inhibit growth, but the lower part with strong auxin transport promote growth, and thus induce bending through the differential cell growth. Okay, and based on these results, my motivation to study plant tropism is slightly different from the previous studies. As you notice some plant behaviors in your town or on your street, the plant shoot has various shapes. For example, in the left panel here, the shoot bends with 90 degrees at a specific location. And in the right panel here, the shoot deflected a little bit and slightly bent at the tip region. And in the bottom, the shoot only deflected downward. So in addition to well-established research field on plant science, where the question is what is trigger or how are the how do signals transport? My research question is as a physicist, it's when and where and how are the forces applied. So what I wanted to do was to understand the various shapes mechanically, which is a problem of posture control of the shoot. Moreover, in order to know the actual shoot morphology, the informatics is needed to quantify the image data. So another possible question is how to quantify the spatio-temporal angle, which can be achieved by solving optimization problem of image analysis. So my recent interest is to combine plant physics and informatics by using interdisciplinary approach to understand the shoot gravitropism. Okay, and this is about data analysis. For the data analysis, we tried to extract the shoot morphology from these image data. To summarize the trials, we first tried to apply the automated extraction by machine learning, but it did not work well because all the images are relatively dark under the light condition, avoiding the effect of light. That means avoiding phototropism. So to extract the morphology, we applied the ImageJ plugin in this study. After some trials, we have found a fast detecting method to extract the curved line like this plotting. And as a result, we could get this color diagram. And this color diagram shows the curvature as a function of time and curvilinear abscissa, which is the arc length along the curve. So you can see the spatial information in x-axis and temporal information in y-axis with curvature in color. And red shows the bending upward and the blue shows bending downward. Through this analysis, we can capture the spatial temporal bending process. For example, the reddish color at the 140 minute corresponds to the maximally bending region around here. Okay, and we then apply this method to the wild type and the lazy worm mutant. In the lazy worm mutant, the gravi-sensing process is normal, but the signal transduction after sensing is somehow disrupted. So it has a relatively weak differential cell growth. To reflect this, you can see that the reddish colors after 140 minutes in the leftmost panel in the lazy worm disappears in the lazy worm mutant in the central panel. Here, I would like to introduce a terminology morphospace, which is often used in morphological or anatomical study. The concept is that only a few characteristic features in morphology are assumed as the representative parameters that distinguish the different shapes or different dynamics. For example, in our case, we picked up the maximum curvature at 140 minutes as a representative curvature parameter and the extension ratio in 140 minutes as a representative growth parameter. As you can see here, we found that the maximum curvature is higher in wild type, but the extension ratio is not significantly different, which are both consistent with the previous work. The point here is that the data analysis itself is not new, but using this morphospace, we try to make correspondence between data and model, which will be introduced next. Okay, and this is about model analysis. Okay, And for the model analysis, we re-implemented the mathematical model of gravitropism proposed by Cherakot et al. In 2017. We call this line vertex model because the plant shoot is assumed as the elastic lot, where the elastic springs are connected with each other at the vertex I. And there are three types of forces applied to vertex I. And the first one is the stretching force, which corresponds to the shoot axial growth. That means that the natural length of elastic spring can extend over time. And the second one is bending force, which corresponds to the differential cell growth. That means that the bending type of elastic spring is connected between the axial elastic spring like this. So the third one is gravity. So all the vertices are under gravity in a downward direction. And the calculation process is as follows. In the current step, the mechanical equilibrium is achieved with the natural length and intrinsic curvature, and then renew the natural length and intrinsic curvature for the next step. And in the next step, the mechanical equilibrium with renewed parameter is achieved. So to slightly bend in this case. Then this process is reiterated to assume the achieved curvature as a current curvature. And to support the consideration, it might be easier with these two equations for natural length and intrinsic curvature. The point here is that the first term in the right-hand side in the second equation correspond to the gravi-sensing, depending on the inclination angle of the shoot. And the second term correspond to the posture control, depending on the curvature. In this case, the current curvature. So in this model, the final morphology can be determined by two dimensionless parameters. The one is bending strength, and the other is elastogravity relativity. Okay, and here is a typical example. And using this model, the shoot gravitropism can be reconstructed like this. And in this movie, the color shows the curvature, and it resembles with the actual shoot morphology. And to make a comment on the first motivation, using the dimensionless parameters, we can modify the shoot dynamics like this. And for example, the light top dynamics with high bending strengths is similar to the first example, this plant. And if the elastogravity relativity becomes high, like this case, the dynamics becomes similar to the second example, like this. And the third one, if the bending strength becomes weak, it becomes left bottom dynamics, and it is similar to the third example. So we can control the shoot morphology in silico and reconstruct the various types of shoot morphology. Okay, and up to here, the analysis were all introduction, and this part is one of the highlights in today's talk. And in order to make correspondence between data and model, we employed the morphospace as a fitting platform. And we searched the best fitted parameters that match the two characteristics between data and model using this morphospace. More precisely, we searched the gravitropic sensing parameter and the relative growth rate parameter in the model. As a result, the data in the circle mark here is fitted by the model in cross mark. As the fitting was done for all the individual shoots, we could get the averaged model for the wild type and the lazy one mutant. The right panels shows the averaged models for wild type and the mutant, where the spatial temporal curvature are qualitatively same and consistent with the actual data. By definition, the maximum curvature is exactly same as the data. By using this method, we could reproduce the averaged bending behaviors of the wild type and the mutant. Okay, and this is main result. And to extract the mechanics of gravitropism, we first obtained the displacement of the averaged model, which indicates how the vertices move in a time interval based on the actual data in this left panel. And importantly, as we have already obtained the mechanical model, we can decompose the displacement into the contributions of stretching and bending forces, like these panels. So the first one is a change of momentum by stretching force, CMS, as shown in these two figures. Surprisingly, it is reasonable that the base of the shoot promote bending, whereas the tip region rather suppresses bending. That means that the shoot axial growth has an opposing effect on bending at the tip region. And the second one is a change of momentum by bending force, CMB, as shown in these two figures. Surprisingly, again, it is understandable that the tip region have a bending behavior and it promotes bending, whereas the base region suppresses bending. So that means that the differential cell growth has an opposite effect on the bending at the base region. So we note that the base region is here and here is under strong applied force, but they are in opposite direction and cancelled out at the base, which results in small displacement at the base. So, and the biological interpretations of this result will be explained in the next slide. And the important point here is in this figure. And we could obtain the force information through making correspondence between data and the model, which is experimentally impossible to observe. So through the mechanical model, we can see the so-called force space, for example, in maximum CMS and maximum CMB in this figure. So, and what I want to say in this slide is that this force space is a new concept beyond the morphospace. And this could be achieved by understanding both data and model simultaneously. Okay. And the interpretation of the stretching and bending forces are as follows. So the stretching force corresponds to the axial change. So the suppression of the bending at the tip can be tip region can be caused by tensile stress derived from the axial growth in this panel. On the other hand, the promotion of bending at the base can be caused by compressive stress derived from the elasticity of the material. So the former is active process and the latter is passive process. And on the other hand, the bending force corresponds to the inclination change. So the promotion of bending at tip can be caused by a gravitropic response derived from log inclination in this panel. On the other hand, the suppression of bending at the base can be caused by proprioceptive response derived from the high inclination in the bottom panels here. So the former is gravitropism and the latter is autotropism or straightening effect. Therefore, the shoot gravitropism can be explained by the combination of active and passive axial change and the combination of the gravitropic and proprioceptive response, resulting in the different applied force. Okay, and to summarize, we extracted the spatial temporal curvature through data analysis and reconstructed bear shape, shoot shape through model analysis. Both analyses are not very new, but the data model combined method is novel and enables us to extract mechanics of the shoot gravitropism. For further investigation, we are thinking about a feedback to biology, especially for mechanics to prediction of differential cell growth. So we are now going to check if some predictions will be hold at the cellular level. To conclude, the take-home message in this study is that considering shoot as a growing elastic rod enables us to understand mechanics of shoot gravitropism. Okay, and for future perspective, further analysis of morphospace is needed to classify different types of bending process, and it will need informatician to objectively determine the morphospace. Also, we are now focusing on the other interesting mutant of posture control, where the myosin 11F and the 11K mutant, which does not have normal straightening ability. Finally, to feel what happens in the model, you can just visit the simulator site here. Although the simulation takes about 10 seconds, so it will be a bit time consuming at the current version, but I hope you enjoy the model world and find some good ideas in there. Okay, and that's all my presentation, and thanks for your attention.
Thank you very much, Satoru. I hope you can hear me. Thank you very much. This was very didactic and very clear, and also very exciting for the plant biomechanics community and the plant modeling community as well. So thank you very much. Maybe I should start with, so I have some questions, but if there are some questions, I think I guess it's either in the chat or Mayra will will tell us. But maybe I can start. So of course, there's some echo with what the team of Bruno Muglia is doing on tropism, and so one of the things that they do is to look at different species with different tropism. So is this? I mean, I guess your model would be applicable to different species, and is it something you want to explore? And
yeah. Exactly, and actually, I didn't explain today, but the model have 6 independent parameters, and I think this depends on the species. So, for example, the initial length or the characteristic length of the shoot is very different between species. So this is one of the answer, and the answer will be, for example, the model will provide different types of bending process depending on initial lengths or other parameters. So maybe it's very interesting to know which parameter affect the species difference.
Yeah, for sure. And I guess you can even extract from your models different parameters for different species. Yeah, no, I understand that. Yeah. And in the same type of extra questions, let's say, if you have you tried to, because all this, when you model or when you do your experiments, the external conditions are constant, right? It's always in the dark, same temperature. So have you ever tried either in model or in experiments to add extra perturbation, like, you know, punctual perturbation or like a fluctuating environment?
Yeah, and actually it's still preliminary, but we are now trying to add different types of stimulation. For example, gravity and light. For example, oscillatively or in a different way, and see what happens to the phototropism, or let's say a combination of gravitropism and phototropism. But it's still preliminary, and we don't know what happens.
For sure, I understand. But since, I mean, it seems like you have a lot of trade-offs happening, since you have like a repressing zone, a stimulating zone. So if you have, in addition, some extra cues, it might be interesting to see. You might have some interesting emerging properties showing up.
Yeah. So, yeah, in my thinking, it needs a lot of improvement of the image analysis, because yeah, I think the behavior becomes complex. yeah, rather than only the gravitropism.
Sure. Yeah,
so
yeah.
It will need some technique to extract the information.
Okay. No, I understand. So we have a few questions. So okay, so Marcos Giacordine says a very nice seminar, and actually there is a question later on, so I'll go to Sadina after. So it's Snapdragon Gravitoprism occurs at the tip. In fact, this is the reason why they are not sold as cut flowers in Europe. I don't know why, because they are shipped horizontally in boxes and arrive all bent up. So this is for Snapdragon. But your seminar indicates that indeed different regions of the shoot do behave differently. So I guess it's more a comment. So I don't know if you've looked into Snapdragon, or I mean, that's what you're saying, right? You talk about the different regions, but maybe you want to comment on this.
Okay, so I'm not sure I understand correctly, but probably Snapdragon is a specific type of plant.
Yeah.
And the bending region is quite different from this type of gravitropism. Is it correct?
Yeah, exactly. That's what he's saying. Yeah? So he says that the Snapdragon is a little bit like exactly the one where you have the pointer right now. The tip, the bending occurs near the tip. And so that's why it's problematic to ship it.
I see. Okay, but
confirms.
In that case, yeah, one possible way is to divide the region and apply the different mathematical model for the different part. And,
so it's like doing a meta model, basically.
Yeah, meta model. Yeah, so it needs model of model.
Yeah, exactly. Okay. There is another question. So more precise, did you look at different ecotypes of Arabidopsis thaliana, and did you see any difference in shoot bending?
Yeah, actually, it's not a systematic experiment, but we tried to see the wild type, for example, Colombia and WS, and see what happens. And if I remember correctly, the bending time interval is slightly different. So in the Colombia, it takes 90 minutes. But in the other ecotype, WS, it takes 100 minutes, or slightly different from the Colombia.
Okay, oh, that's interesting.
Yeah.
There's also an interesting ecotype to look at, I guess, would be Landsberg erecta, because it has very, stiff stems. So I don't know. Could be interesting. Okay, there is another question. Do plant cells, so from Josapol Arnivanishvesh, sorry, I don't know if I pronounced it correctly. So I know that Josapol is from Dolphiger's lab. Do plant cell wall mutants show different pattern of curvature and extent ratio? Have you looked at cell wall mutants?
Cell wall mutant. Okay, actually, we are going to see such mutant. But one interesting experiment so far was the, let's say, in the posture control mutant, and we tried to see the Young's modulus of the shoot, and they are not very different from the wild type. So this bending process, this time course is from top to bottom, and this very bending shoot have almost same Young's modulus as a wild type. So in this case, the cell wall it does not affect very much on the bending process. But yeah, we don't know yet about the cell wall mutant.
Okay, yeah. I'm thinking of one, of course, the Catani mutant would be an interesting one to look at.
Okay, Catani mutant.
That's so brittle stems, right? So it could be a good one.
Yeah. And the microtubule will affect this process, I think.
Yeah. I guess so. And there's a second question from Josapol, but I guess you already answered it to some extent. Can you estimate Young modulus value of plant cells based on your model?
Okay. So. Yeah, it's very interesting and important comment. And for example, at the moment, we try to estimate the Young's modulus and also bending modulus in this case. And actually, we get some numbers, Young's modulus, but it explains the elasticity of the material. But in our model, this is an active process, so it includes growth of the material. So the Young's modulus as the elasticity does not make sense to the active process. So in my opinion, it needs two types of stiffness will need to be considered. So one is elasticity, so with a static shoot, and one is growing, how to say, growing property of the shoot. So yeah, I'm thinking about that thing.
Yeah, no, exactly. So I think you're right. It's a mix of elastic and plastic behavior. So it's like more extensibility in some ways rather than pure. And I guess at which scale can you deduce your properties? So when you, in your model, it's integrated over several cells, of course, right? So it's over like it's a region of the stem, right? So you can't have a resolution at a single cell level, right? Or at least
exactly, yes. And this model is organ level simulation. So if we want to know the cellular level dynamics, it needs more, how to say, fine structured model with cellular resolution. But it will probably possible with the finite element method or some other method to see what happens.
Yeah. And speaking, oh, sorry. So speaking of scale, I was thinking of the temporal scale. So here you show the movie where you have something slightly continuous, I would say, like a movement that is average over time. Do you detect either in data or in model some small fluctuation in the movement?
Yeah, actually, yeah, it's also very important comment. And yeah, for example, yeah, let's move on to this slide. And for example, if you say about fluctuation as a total length or actual length, it sometimes fluctuates. For example, with a 50 minutes or to 80 minutes oscillation behavior. And we found that this is come from the nutation, circumnutation effect. So if the image is not captured from the exactly vertical axis, or how to say, perpendicular to screen in this case, it sometimes capture the circumnutation. So yeah, it sometimes fluctuate in the total length. But is it the answer to you?
Yeah, no, exactly. But I'm thinking that this could also be an interesting cue, right? Because those movements are also probably contributing to the overall response. I mean, it's movement in 3D, but it could, influence the 2D behavior, right? In the end.
Yeah.
Like the curvature.
Yeah, and one caution in the statement is that in this case, the human manipulation or human data analysis include some error. So
yeah, of course.
yeah. So you can find some fluctuations like this here, but it sometimes the human detecting error. So it's not the fluctuation of the system.
Right.
So we need to take care of such data analysis. Yeah.
It's a fluctuating human eye.
Yeah.
Okay, so I don't see more specific questions. So maybe I can ask maybe a last general question for the audience, because since we are a journal that is all about interdisciplinary connections and bringing people from different fields around plant science. Maybe I can ask you, so since you're coming from physics or statistical mechanics, as you said, how difficult was it to move into plants, and what would be your advice for a physicist who would be interested in joining the plant science community? It's a broad question.
Okay, so it's a bit difficult question, but yeah, in my thinking, what we can come together should be the data analysis. For example, for experimentalists, data analysis will be needed to understand the data, and for physicists, it is somehow the milestone or trigger to study plant with the data analysis. So, yeah, one of the platform or gathering point might be the data analysis, and so my how to say, my hope is that the many physicists can attach or can encounter many data and see what happens in the data analysis first, and then think about physics later. So maybe that is one possible way to come together. Yeah, between plant science and the physics society. Yeah,
I think it's a good point actually, because it's true that we can provide a lot of material to analyze. So it's a good point. Actually, I have a colleague in physics who said that he was tired of physics system that were too simple, and that plant science is a truly complex system. Really complex. Okay, well, thank you again. So I think I'll hand to Myra to conclude the seminar.
Thank you, Olivier, and thank you, Satoru. If there are no other questions to come in, I hope this Q&A has covered and answered everything you might have had. So I really hope you enjoyed this webinar and found it useful to hear from Satoru and Olivier and learn more about the journal Quantitative Plant Biology. I'd really like to thank our panelists and the team for their time and for taking part. There are more webinars in this series, so please kindly see if you'd like to register for the other guest speakers that we do have lined up. And I'd just like to thank you all very much for attending. So thank you so much, Satoru, Olivier, Richard and Ali. This has been a very brilliant session. Thank you.
Thank you very much, Satoru. Thank you, Myra.
Thank you very much.
Thank you.
Bye-bye.